Good afternoon. How are you? Am I audible to you? Hello.
Assalamu alaikum, sir. Yes, sir. Good afternoon.
Good afternoon. Am I audible to you clearly?
Yes, sir. Yes, sir.
Okay, then we shall start our talk. Can you see the slide?
So we can, sir. Sir, I have an information to give, sir. Sir,
Yes.
one of the student, Abrar, he.
Yes. What happened to Abrar?
Yes, sir.
What happened to Abrar?
Sir, one of his family members is hospitalized. So, sir, he informed that he could not be able to join today's class.
All right. Thank you. And let us wish for his family members. Today, we are going to start on terpenoids, the most important class of secondary metabolites. There are different classes of terpenoids. For example, hemiterpenoids or hemiterpenes, monoterpenes, sesquiterpenes, diterpenes, triterpenes, tetraterpenes, or polyterpenes. You can see the example in case of hemiterpene, isoprene is the example. This is the smallest terpene because terpenoidal compounds, they contain carbons 5, 10, 15, 20, 30, like that. So the smallest unit is C5, that is carbon in the structure, at least 5 carbons are present. Isoprene is the best example for hemiterpenes. Monoterpenes, limonene, sesquiterpene, abscisic acid, very famous phytohormone. Diterpenes, gibberellin is another important phytohormone. Triterpene, brasinosteroids. Brasinosteroids are also important compound which act like phytohormones, hormonal compound in plants, as well as involved in different signaling pathways. Tetraterpenes, carotenoids, in the carrot, carotenoids are important for a source of vitamin A for us. For example, beta carotene. Polyterpenes, for example, rubber, or ubiquinones. Ubiquinones or rubber, mixed biosynthetic origin. There are some meroterpenes also. For example, cytokinin, vitamin A. Their biosynthetic origin is a bit different than the classical terpenoids, a bit mixed. So terpenoid, if you ask to classify terpenoid, then you need to list and with example up to the meroterpenes. So these are the some examples of monoterpenes. For example, linalool or menthol in the mentha, linalool possibly found in the lemon, and there are many others, and all are aromatic compound. Sesquiterpene, these are the sesquiterpene, how they are biosynthesized, diterpene, and triterpene, lots of compounds. No need to memorize all, but you need to at least know at least one classical compound for each class. Tetraterpene, carotenoids, very rich Carrot is very rich in carotenoids. These are the brief description, classification of the terpenoids. And terpenoids are very important group of ecochemicals. They play a vital role as they in communication of the producer, as most of them, many of them are volatile. Some of them have the hormonal functions in plants and other organisms. For example, steroids are also like that. Another very important class of secondary metabolite, as well as the ecochemicals, are flavonoids. Flavonoids are basically phenolic compound. Flavonoids have a general structure also for information. They are polyphenolic compounds found in softwoods and hardwoods and many other parts of the plant, composed of diphenyl propane units. Here you can see there is a skeleton of flavonoids, C6, C3, C6. That means A ring is C6, B ring is C3, carbon 3, 1, 2, 3, and C ring, and B ring again C6. Here, interestingly, you see numbering. Numbering starts from here. 1, 2, 3, 4, 5, and 8. And B ring is again 1 prime, 2 prime, 3 prime. So this is the classical method for numbering the carbon of in the flavonoid structure. That means composed of diphenyl propane unit, C6, C3, and C6. This is applicable for the classical flavonoids, but flavonoids can be branched and other position may be substituted. They have 8 structural classifications. Flavonoids differ through substitutions on the rings. Typical OH or OCH3 groups. Many of the carbon hydrogen is substituted with hydroxyl or methoxyl group. Typical to have position 5 position, 7 position, or 3 prime position, 4 prime position, even 5 position. These are the typical position where substitution is taken place, either mainly OH or even OCH3, that is methoxyl group or hydroxyl group, are also present as glycoside. This is very important. What does it mean? Some of the substitution, for example, 5 prime can be 5 position, this hydrogen can be substituted with the glucose. So glucose can be connected to the structure, then they are known as glycosides. And if there is no, glucose, the structure is called aglycone. Aglycone means no side chain, big side chain, or like that. This is very important classification of the flavonoids. Here you can see the structural differences. The first group is called flavones. Flavones means, here you see double bond, here double bond. Flavones are not likely, this is the flavone skeleton, but there might be substitution in other position, but this is the critical. Double bond here and double bond here. This is the compounds having these 2 double bond are known as flavones. The second group is flavonols. Flavonols means, here you can see all that is hydroxyl is added. So these 2 structures are very close, only here hydrogen is substituted with hydroxyl. And then flavonones, there should be one N, N-O-N-E-S, flavonones. Here you can see no unsaturation here. That means 2, 3 carbon, no unsaturation. This is why flavonones. And flavonols means flavonones with hydroxyl group. That means an all group is added. And other group is catechin. Catechin, here you see no oxygenation in the 3 carbon. So it is catechins. Tea, we are drinking tea. Tea is rich in catechin. Leucoanthocyanidins. Leucoanthocyanidins like catechin, but 2 hydroxyl group in the 3 and 4 carbon. Chalcones, another group. Chalcones, here you see some peculiarities. Here, this oxygenation, this part is open, and here is the double bond, here is the double bond. So chalcones. And another one is oruns. Oruns means double bond here remain, but here you see structure is peculiar and change. And there is another double bond here. And this structure, this structural group is called oruns. So total 8 groups of flavonoids are abundant in the nature. For example, leucoanthocyanidin or anthocyanidins are the color of plant leaves and flowers and abundant in the nature. Representative structures of flavonoid classes, for example, chrysin, chrysin flavones, flavonols, or you can find many other representative structure, for example, texifolin, pinosembrin, pinobunskin, catechin, western hemlock, or quercetin, or oak. So these are some examples of a natural compound. Structure of flavonoids, here you can see how there are changes. For example, I told you this carbon is 2, and 3, 4, 5, 6, 7, 8, and then here 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, 6 prime. So here hydroxylation is common, but it can be methoxylated, or other structure changes can be happen here. Either a hydroxyl group or methoxy group or other group can be connected. Even I told you the glucose molecule can be connected. Even one more glucose can be connected in a chain. And similarly, these two, 3 prime, 5 prime also can be substituted. Generally, 4 primes substituted with oxygen. Here you can see 3 flavonols. 3 flavonols means camphorol, quercetin, myrcetin. That they are very close in structure. This is the camphorol. Closely look at. Here you see 3 hydroxyl and 5 hydroxyl, 7 hydroxyl, and here 4 prime hydroxyl, and all are classical flavonol. Then here you can see quercetin, just 1 more hydroxyl in the 5 prime position, and then another 3 prime position hydroxyl myrcetin. So these are the structural variabilities, and due to the structural variabilities, they are functionally also diverse. So structure of flavonoids, quercetin is yellow, and this as and color also are very different. Catechin is white, and cyanidin is red. Cyanidin is the pigment present in the flowers and even in the leaves. Most of the flowers, red flowers, have cyanidin. So here you can see pelargonidin, cyanidin, delphinidin, rose color, red color rose contains cyanidin, and pelargonium, for example, geranium contain pelargonidin, and different. These are generally called anthocyanin pigments, delphinidin. So these are classical examples of the pigment present in the flower. And how they are biosynthesized? Mostly, flavonoids are biosynthesized through the shikimic acid pathway. You see pentose phosphate pathway. There is a pathway, very famous pathway in the cell. Glycolysis also another. So here, phosphoenol pyruvate and erythrose 4-phosphate can change into shikimic acid. And from shikimic acid, you see all the different kinds of secondary metabolites, mainly flavonoids, can be biosynthesized. And flavonoids, mainly phenyl alanine lyase, from shikimic acid to phenylalanine can be produced, and then phenylalanine lyase can be involved in the synthesis of the flavonoids, lignins, and many other compounds, coumarins. And flavonoids can be many units, not monoflavonoid only, can be polyflavonoids. Polyflavonoids are called the condensed tannin. And another, gallic acid, they are also highly phenolic compound, gallic acid. Gallic acid also found in the plant abundantly, even in the tea, so they are hydrolyzable. That tannin, they can be hydrolyzed. Shikimic acid pathway also generate tryptophan and other amino acid, and they can be used to derive different alkaloids. So there are many diversity of compounds, and as I mentioned before, the secondary metabolites are derived from the primary metabolites. So this is all about today's brief talk, and if you have any question, then it would be interesting. So you can ask some question, and then I shall discuss about your exam, because you need you receive lots of lectures. You need to have a midterm exam or quiz exam. So you can ask some question now. This is your turn. Please ask some question. One member may be disconnected. Merjabin Mim, do you have any question?
Sir, not exactly question. Sir, the pathways are a bit complicated. So, sir, it's a bit confusing.
Yes, pathways are complicated, but imagine how scientists discovered this pathway in the cell. Really exciting. So pathway, if you concentrate, it would not be complicated. This subject possibly is new because in our university, the curricula is, almost free from the chemistry. But chemistry is life. Everything you see around you, all are the chemicals. Your life is chemicals, DNA, RNA, everything. But here chemistry is very poor. Whatever you see is chemistry. People study the pollination, but even they do not know the how pollination is taken place. Just flower like lemon, insect comes, and why insect comes, and what are the communication mechanism. These are the important. So biosynthetic pathway, first time it looks complicated, but if you go through carefully, then you can easily understand. You studied the TCA cycle, glycolytic pathway, and many others pathway in biochemistry or plant physiology. These are like that metabolic pathway. They are obviously complicated. I am showing you only the.
Sir, I have an interest. How these mechanisms are discovered?
How? Pardon? How?
And first to know how these mechanisms are exactly discovered, like this pathway,
Yes.
this chemical here, how this
Yes,
is
this is why
happening.
you need to know the chemistry.
happening.
Yes, you need to know
Sir, how these mechanisms are discovered by the scientists, as these are much more complicated.
yes, it was not discovered by a single man. Thousands
So
of
the network might be a bit
thousands of scientists discovered the whole story piece by piece. So someone got some compound, another got the compound, how the structural relationship, and then they discovered the enzyme. And there are some, chemical formula I told you from one flavonoid to another flavonoid. So it is not discovered just by a single scientist. One fine morning, it was not discovered. It was all the pathways, even lots of things are still unknown. All the pathways were discovered by, the discovery of thousands of scientists. First, scientists discovered the chemicals. What are the chemicals present there? There is a way you can extract the organic compound from any materials by using organic solvent. And then once you get the compound in the solution, then you can purify by using chromatographic technique. I shall discuss later on. And after purification, then there is a challenge to elucidate the structure. What is the chemical structure of this compound and the biological activity? So all the things are very challenging, but scientists piece by piece, discovered the complete story, but yet many stories are unknown. So you can discover in future. Any discovery, fundamental discovery is exciting and simple once you discover, but very complicated. It's a darkened world. Any more question? I saw many of you are unstable in internet connection. Kashfi, are you there?
Yes, sir. Sir, we are all in lab and the network is
Yeah,
very unstable.
network
They
is.
can't even connect.
When is the tentative schedule of your first midterm?
Sir, this is the second week, so maybe fourth week.
Fourth week. So we can even take midterm exam next week. Next
week.
Yes. Next week, because you receive lots of interesting topics. And the next class, be prepared to have a quiz exam. Rojana, are you connected? All are unstable. Rojana.
So she is here.
But
Sir,
I can, yeah.
Sir, I can't connect through my phone actually. So the only Kashfi is connected now.
Okay.
We all are good here.
Do you have any question or comment?
Sir, actually, I didn't get any single point of this today's lecture. So
Okay.
the recording is very essential, necessary, sir, for us.
Actually, we discussed today the different classes of terpenoids and also different classes of flavonoids. And I discussed some examples of terpenoids and flavonoids. That's all, not so very complicated. Actually, the flavonoids and terpenoids, they are very important in the point of economy as well as life. Many medicines are derived or discovered from these two classes of compounds. And in agriculture, they are very important. Flower colors flower aroma, or different phytohormones. They are all belonging to flavonoids and terpenoids. And flavonoids are very important in plant defense, plant communication. Terpenoids are also. So to understand the molecular mechanism of plant defense, plant communication, not only plant, even other organisms also have the terpenoids and flavonoids. Naz Hassan, are you connected at this
Yes, sir.
moment? Have you received the lecture uninterruptedly?
Ji, sir.
Okay, good. Do you have any question?
No, sir. All sir structure.
Okay. I have already shared the PowerPoint slide that is in PDF format, the secondary metabolism of plants. And I shall share the video lecture also. Tasneem, are you getting the lecture or connected, disconnected in the mixed situation? Anyway, thank you very much. And I shall record I am recording the lecture, and you can get the whole story, and you can have the lecture in the YouTube. Thank you so much.